Hello, today we'll be discussing the article Excess of Genomic Defects in a Woolly Mammoth on Wrangel Island, authored by Rebecca L. Rogers and Montgomery Slatkin. During the Pleistocene and early Holocene period, Mammuthus primigenius, aka the woolly mammoth, populated Siberia, Beringia, and North America. Recently, two woolly mammoths from two different populations, but of the same species, have had their DNA sequenced. One is from a mainland population and was found at Oyakhon in Siberia. It is about 45,000 years old. The other is from an isolated population on Wrangel Island and is 4,300 years old. The mainland group of mammoths would have had an effective population of almost 43 times larger than the Wrangel Island group. These two populations offer a chance to examine how the genome responds to pre-extinction population dynamics. Because the mammoths are the same species, there are no biological variations that would be present in a cross-species comparison. Since the Wrangel Island mammoths had such a small effective population size, it was hypothesized that the mammoths from Wrangel Island would have had more mutations, such as retrogenes, deletions, and premature stop codons. This was tested by comparing the genomes of the two mammoths.
There are multiple pieces of data that demonstrate genomic decline in the small population of the Wrangel Island mammoths. The first piece of data is the amount of deletions found in the Wrangel Island genome versus the Omiacon genome. Over 1 kilobase pair, there are approximately 21,000 deletion mutations in the Omiacon genome, whereas there are approximately 27,000 deletions in the Wrangel Island genome. Additionally, there are far more exons deleted in the Wrangel Island genome than the Omiacon genome. There are 1,100 genes with exons deleted in the Omiacon genome, and there are 1,600 genes with exons deleted in the Wrangel Island genome. Another piece of substantial evidence comes from the amount of retrogenes in the mammoth genomes. There are 1.3 times as more retrogenes in the Wrangel Island mammoth than in the Omiacon mammoth. While retrogenes are not inherently negative, they may point to a burst of transposable element activity in the lineage of the mammoths. This transposable element activity would have likely had a negative effect on the individuals of the mammoth population. The last striking piece of evidence is the amount of stop codons found in the Wrangel genome. There are 1.8 times as many premature stop codons in the Wrangel Island genome than in the Omiacon genome. Additionally, there are more errors in the stop codons of the Wrangel Island genome. There are 377 genes truncated in the Omiacon population, whereas a whopping 645 truncated genes in the Wrangel Island genome.
The main results, confirmed by this experiment, revealed that the Wrangel Island mammoth had an excess of amino acid substitutions and stop codons as compared to the Omiacon mammoth. These changes, likely a result of shrinking population size, led to loss of olfactory receptors and urinary proteins, as well as gain of a satin coat phenotype among the island mammoths. This example of genetic relationship is especially important on a grand scale for multiple reasons. One reason that this study is different from previous experiments lies in the intense difference in population size within a single species. This variance allowed scientists to examine the evolution of the genomic architecture of woolly mammoths. Another way that the information regarding the mammoths can be more widely applied is through varying genomic defects in different species. Some of the genes deleted in the mammoths from Wrangel Island, while they did not lead to fatality in the mammoths, could have led to fatality or more important phenotypic changes had they been evolved in other species. For example, riboflavin kinase, which was deleted from the mammoth, leads to fatality when deleted in mice.
Now the question becomes, how does this journal article link to what we have learned in Bio 1510? First of all, it could be linked to Module 2, Ecology. Remember how there are 5 levels of ecology? This particular journal article deals with the 4th level of the ecosystem since it is about the interaction between the environment and the species. The extinction of the mammoth was largely due to climate change according to the journal article. Also, remember the theory of island biogeography? It stated that the more isolated the island, the immigration rate is low while the extinction rate is high. Since the Wrangel Island was isolated and it had low immigration and high extinction rate, we could assume that the island mammoth population was susceptible to more mutation and genome deterioration. Now, let's go back to Module 1. In Module 1, we learned about the Founder's Effect. The Founder's Effect is that the genetic diversity is restricted since the diversity is only from the few ancestors that moved to the island from the mainland. Moreover, this article reflects that mechanism of evolution. We learned that when the population is small, it is more subjected to mutation and genetic drift. Thus, this could explain why the Wrangel Island population had a higher mutated gene as compared to the mainland population. Lastly, we will move on to Module 3. A stop codon is a triplet of amino acid that is embedded in mRNA. It tells us when to stop synthesizing protein. The Wrangel mammoths had a premature stop codon which affected the proton's secondary, tertiary, and quaternary structure.